Sophie Levi,
Addison Mueller,
Palvi Maharaj,
Jenna, Roark.
Dolly's birth blew the world away. Scientists had taken a single adult cell from a sheep's udder, implanted it into an egg cell that had been stripped of its own DNA, and successfully created a living, breathing animal, almost genetically identical to its donor. This is a quote from Joanna Klein, a New York Times journalist who graduated from New York University's Science, Health, and Reporting program, published in the article Dolly the Sheep's Fellow Clones, Enjoying Their Golden Years, written July 26, 2016.
We have all learned about cloning in 10th grade biology with Miss Heitman.
Cloning is a complex science found within research labs as well as nature.
First, we will discuss what cloning is. Second, we will talk about the 3 types of cloning. Third, we will talk about and explain animal cloning. And lastly, we will discuss plant cloning. Let's begin by taking a look at the history behind cloning.
Dr. Goyle, a Canadian oncologist at the Edmonton Hospital, stated in a personal interview on December 20, 2016 that cloning is a replication of genes. This means that every piece of DNA is exactly the same between the cloned organism and the original organism, which causes them to have the same features, like facial features. Scientists are using cloning to make research easier. The goal of cloning is to make organ donation 10 times easier in the future. Bryce McConney, a college student at the University of Florida and Haiti journalist for Medical Daily, wrote A History of Cloning Before Dolly the Sheep on July 12, 2014 that cloning began before the 1990s by a scientist named Robert Briggs at the University of Utah. He and his partner Thomas King were the first scientists to successfully start cloning after using nuclear transfer on experimenting on frogs. Nuclear transfer is a form of cloning. Cloning is technically known as somatic cell nuclear transfer, or SCNT. Dr. Subramaniam, a scientist and researcher at the Mayo Clinic, stated in a personal interview on December 28, 2016, the process of cloning. Dr. Subramaniam states that first, scientists impregnate the donor sheep through a process called IVF, or in vitro fertilization. After the sheep is pregnant, the scientist will extract the sheep's skin tissue by doing a skin cell biopsy, which is a procedure of removing the skin's specialized cell and examining it under the microscope. If the tissue is healthy, the scientist will continue by destroying the nucleus and fusing it with an unfertilized egg of another sheep. Second, the unfertilized egg is zapped with electricity until it becomes a blastocyte, which is where they start to form a fertilized egg. Thirdly, after the egg is formed, the fertilized egg is then implanted back into the second sheep, or the surrogate sheep, and resulting after 147 days is when the newborn sheep is born, where the newborn sheep will have the identical genetic replica to the donor sheep. Most importantly, this showed that nuclear transfer is a viable cloning technique, as stated in Dr. Subramaniam's article. However, Vanessa Rose, a college student at the University of Minnesota and a physicist researcher, stated in a personal interview on December 28, 2016, that there are a few types of cloning. Now that we know how cloning works, let's look at the 3 types of cloning.
Molecular, therapeutic, and reproductive are the 3 kinds of artificial cloning. The first type of cloning is molecular cloning, also known as gene cloning, which is when just one gene is cloned, as opposed to an entire cell or even a whole animal. A gene is a of our DNA that codes for just one thing our body does. And according to the National Human Genome Research Institute, which successfully mapped all genes of the human genome, in an article titled Cloning Fact Sheet, published May 11th, 2016, the process for molecular cloning is much different than the process for therapeutic or reproductive cloning. First, as you can see, all the genes are extracted from a cell. This is so that scientists can find the specific gene they are looking for. Then, once the gene is found, it is implanted into another cell, and that cell starts to display certain characteristics of the first cell. Molecular cloning is useful for scientists in many ways. It first allows them to work out the function of a gene. This is because they can see what it does to an organism that it has been added to. Also, scientists can see how mutations may affect genes as well. And finally, scientists can add certain traits that they want to organisms. An example of this is golden rice, which has been fortified with vitamin A by gene cloning. Vitamin A is essential for our vision, and many children in third world countries do not get enough of it. Golden rice helps prevent these children from going blind due to a vitamin A deficiency. Another example of scientists adding traits to organisms is insulin. According to Laura Fraser, a New York Times journal, author of 3 New York Times bestseller scientific nonfiction books, in the article Cloning Insulin, written in 2015, the insulin for diabetics used to come from animals, but the insulin from over 23,000 animals had to be collected for just 1 pound of useful insulin. Scientists wanted to change this, so they added the gene for insulin production into bacteria. Today, all of the insulin that diabetics need can be produced in the lab, and no animals have to die. The second type of artificial cloning is therapeutic cloning, which involves using somatic cell nuclear transfer, or SCNT, to clone tissues. These tissues are grown in a lab, and they are used for scientific research. An example of this is that scientists can take cells and clone them from someone with a disease, and then that disease can be studied without ever having to test on humans. Currently, scientists are trying to figure out how to make the cells cloned using therapeutic cloning differentiate or transform into other types of cells. This could eventually allow them to clone entire organs, and those organs would be used to help patients who are waiting for an organ transplant. Typically, they have to wait for a donor, and many die before they get an organ, or die because they rejected an organ. According to the U.S. Department of Health and Human Services, in an article titled The Organ Transplant Process, written January 29th, 2015, 22 people die each day in the United States because they did not receive an organ on time. And according to the Mayo Clinic, the top-ranking hospital in the world, in an article titled Results, published June 24th, 2016, 14% of kidney transplants are rejected after the first 5 years. Both of these numbers could be significantly reduced thanks to therapeutic cloning. First, patients would not have to wait for an organ transplant because they could grow their own organ. And also, most of the time, organs are rejected because your body attacks foreign cells. But this way, the cells would not be foreign. They would be your own, so your body would not attack them. The final type of artificial cloning is reproductive cloning, which involves cloning entire animals using somatic cell nuclear transfer. This type of cloning could be very useful for couples who are having fertility issues. According to Robert Sparrow, professor at the Monash Bioethics Center, in a peer-reviewed article titled Therapeutic Cloning and Reproductive Liberty, published in the Journal of Medicine and Philosophy in 2009, this would work in one of 2 ways. The first way is that DNA from a father would be inserted into an egg with no nucleus from the mother. A child created this way would be genetically like a twin to their father. And the second way that it will work is by using DNA from an existing child of the parents, or by using stored tissue from a deceased child. A child created this way would be a clone or like a twin to the parent's existing child or to the deceased child. This type of it is good for scientists to have as few variables as possible. And this type of cloning allows scientists to produce many genetically identical mice, therefore reducing variables.
Next, let's take a closer look at some of the animals scientists have cloned using the reproductive method. Scientists have been attempting to clone a variety of animals for years. In the year 2001, scientists attempted to clone a gar. The gar is the largest existing species of cattle in the world. For this experiment, scientists removed the nucleus from the skin cell taken from a garble who had died 8 years earlier and fused it with a regular cow egg. According to Oliver Tickle, an author of 3 scientific books and an editor for The Ecologist, in his article titled Noah's Ark Revisited, which was posted in March of 2001, of the 692 eggs used in the experiment, only 1 calf was born. The gar calf, who scientists decided to name Noah, showed symptoms of a common infection just 1 day after being born. Due to this infection, Noah only survived 2 days. Another animal scientists have cloned is the Pyrenean ibex, or bucardo, which is a type of wild goat. Celia, the last remaining bucardo in the entire world, was crushed and killed by a falling tree in the year 2000. However, scientists were able to preserve some of her cells after her death with the intent to clone her. Of the 57 surrogate goats used in the experiment, only 7 became pregnant. Of those 7, 6 had miscarriages. Only 1 mother carried to term, and she gave birth to Celia's clone. Sadly, Celia's clone had a lung defect at birth, and she survived only 10 minutes. Not all clones have died so quickly, though. A few years before Celia and Noah, in July of 1996, Dolly, a cloned Finn Dorset sheep, was born at the Roslin Institute in Edinburgh, Scotland. According to Teresa Wimmer, an author of over 30 scientific books, in her book, Cloning Dolly the Sheep, which was published July 1st of 2008, stated that Dolly the sheep was named after Dolly Parton, an American country singer, because Dolly the sheep's DNA came from a mammary gland cell. The team behind Dolly was headed by Ian Wilmot and Keith Campbell. 1 year earlier, Wilmot and Campbell had cloned 2 other sheep, named Megan and Morag, from 9-day-old embryos. What made Dolly different from Megan and Morag was the fact that she had been cloned using adult sheep cells. For Dolly's experiment, scientists took a donor cell from a Scottish black-faced sheep and removed the nucleus. They then fused the empty cell with the nucleus from a white Finn Dorset sheep. Then, the scientists replaced the fused cell into a surrogate ewe. And so, Dolly, a white Finn Dorset sheep, was born to a Scottish black-faced sheep. In February of 1997, Dolly's presence was announced to the world. Dolly lived a relatively normal life for a sheep, minus the media coverage. According to Mark Westison, a professor of veterinary physiology and pharmacology at the Texas A&M University, and Jorge Piedrohita, a professor of translational medicine at the North Carolina University, in their article titled 3 Little Pigs Worth a Huff and Puff, which was posted in November of 2000, that Dolly was able to breed normally, and she gave birth to 6 healthy, normal lambs throughout her lifetime. However, in 2002, Dolly developed arthritis. Arthritis is not uncommon in sheep once they hit 10 years old, but Dolly was only 5 when she developed arthritis. Dolly the sheep ended up passing away just 1 year later. According to Ewan Calloway, a senior reporter for Nature, the most cited scientific journal, in his article titled Dolly at 20, the story behind the world's most famous sheep, posted on June 26, 2016, that Dolly's death, which was caused by lung disease, was not related to her being a clone, although her arthritis may have been. Dolly was only 6 and a half when she died, which is half the average lifespan of a Finn Dorset sheep. After Dolly developed arthritis, Wilmot and his team began to research the impact cloning has on animal health, because they feared that Dolly's arthritis may have occurred because of a genetic defect caused by the cloning process.
Now that we understand animal cloning, let's take a look at plant cloning. According to Dr. Julie Hayes, a research associate for Australian Centre for Plant Function, in an article titled Cloning Plants, published in the year 2008, Nature has been cloning plants for billions of years. Plants make identical copies of themselves by asexual reproduction. According to Merriam-Webster Dictionary, updated in 2016, asexual reproduction is reproduction without the union of individuals or gametes. In simpler words, asexual reproduction is reproduction when an organism comes from a single parent plant. Plants either clone themselves by tubers or runners. Tubers are grown at the roots of the plants and store nutrients, energy, and DNA in them. Some examples of tubers are potatoes and sweet potatoes, also known as red tubers. Runners are root-like and grow along the soil Runners are root-like and grow on the soil to form a completely new plant that is genetically the same as the old plants. Some examples of runners are strawberries, mint, and grass. According to Matt McGraw, an environment correspondent for BBC News, in an article titled Cloning Plants, published in August of 2005, plants can also be cloned by using a technique called vegetation propagation or cutting. Cutting is the simplest way that plants can be cloned, and we have been cutting plants since the beginning of agriculture. Vegetation propagation works because it forms a mass of non-specialized cells called callus. When doing the cuttings on plants, gardeners often cut the plants in branches or bulbs and keep them in a warm, moist environment for up to 2 weeks until the new plant starts to grow.
In conclusion, first we talked about what cloning is and how it works. Second, we discussed 3 different types of cloning.
Third, we explained animal cloning, and lastly, we went over plant cloning.
Cloning is a complex science found within research labs as well as nature.
While Dolly's birth was revolutionary, we have many more exciting things to look forward to in the future of cloning.
17